Well, thanks everyone for coming here today, and thank you, Zoran, for participating in this webinar. It looks like we have a large amount of people that have joined, so thanks and welcome everyone. Excited to have you all here. But yeah, I'd like to welcome Professor Zoran Radic, who's going to be presenting this webinar with us today. Professor Zoran Radic is an adjunct professor at UC San Diego in the Skaggs School of Pharmacy and Pharmaceutical Sciences. Professor Radic is a structural biologist and biochemist working on the field of cholinesterases. He actually graduated from the University of Zagreb in Croatia, where he started his research on acetylcholinesterase. As a postdoc, he joined the lab of Dr. Palmer Taylor at UCSD, where he contributed to the functional mapping of the acetylcholinesterase enzyme. Currently, in the capacity of an independent faculty member, his research focuses on finding antidotes to treat organophosphate poisoning, guided by the structure of acetylcholinesterase template. He has been a big VR supporter over the years and provided key feedback, really since the early days here at Nanome. I remember going into his office and showing him all of our virtual reality stuff and really closing the deal and like having him really be our first big customer using our product for molecular design way back in 2017. So yeah, thanks so much, Zoran, for helping support Nanome and support everything we're doing, providing your feedback, as well as thank you for your research. Really has a real world impact, as we will all learn here shortly. But yeah, without much further ado, I'll let you take it from here.
Thank you, Steve, and thank you for the software that we are all able to enjoy now at a fairly advanced stage. This is Zoran Radic speaking from UC San Diego, Skaggs School of Pharmacy. And this is enzyme that I've been studying for quite a few years, actually decades now. It is acetylcholinesterase, a key enzyme in cholinergic neurotransmission. This is a catalytic subunit of it with active center buried about 20 angstroms deep in the center of this globular molecule. And that's where the active serine, the most important catalytic residue, resides. But what you see here at the base of the gorge now is not the serine, it is a covalent inhibitor of enzyme. It is organophosphate, and its structural formula is in the lower right corner. This is one of most toxic compounds ever made by man. This particular one falls into category of Novichok nerve agents that have been accepted or enlisted into very short list of prohibited chemical warfare by Organization of Prohibition of Chemical Warfare only last year. So we are interested in recovering activity of enzyme inhibited by this type of inhibitors. It's not only nerve agents, it's also organophosphorus-based pesticides that cause poisoning by this type, covalent poisoning of this enzyme, and malfunction of cholinergic neurotransmission. So we are in a field of designing, based on enzyme structure, designing antidotes against organophosphate poisoning that are supposed to enter active center cavity of this inhibited enzyme, attack phosphorus, and recover catalytic activity. One of candidate antidotes you can see on the left in this little movie that I recorded in VR. But before going into molecular world, let me remind you of few cases, high-profile cases of poisoning by organophosphates that happened in recent years. Those were featured in major communications media like CNN. So let's move to CNN and their coverage of the poisoning. But before getting to poisoning, let me remind you that Nanome and its software has been featured recently, and this is CNN feature of Nanome just a few weeks ago. So the poisoning case that we now study using this software, in addition to other tools, was of a major opposition figure in Russia, Alexei Navalny. It happened in summer of 2020, in August. And the photograph that you see in the left is Alexei sitting at the airport of Russian town Tomsk in the morning of that day, waiting for the airplane. At that point, he was already poisoned by one of three compounds listed here in the yellow rectangle in the lower part of the slide. He was exposed in his hotel room, most likely, and reportedly one of those compounds was applied to his underwear in the form of powder. That is one of unique features of this new class of nerve agents that, unlike other nerve agents that are called sometimes nerve gases, are very volatile. These are not. These can be prepared as solids and are therefore much more stable, about 2 orders of magnitude more stable than sarin, for example. And that's the way their hazardous properties come from. So the serious symptoms of poisoning in this case came 1 hour later after this photograph was taken, when he was already in the air. Unfortunately, pilot reacted very quickly and promptly and landed the commercial flight to nearest airport in Omsk. Alexei was taken to ICU unit of local hospital and treated there, but 2 days later he was transferred to Germany. So what happened, and then here in the photograph on the right, you see him after, approximately 1 month afterwards, partially recovered. So treatment initial in Russia and then later in Germany saved his life, and we are interested in treatment of that kind of cases. But first, what those poisons did to his system and at the molecular level. Those are really nasty compounds, and we don't know much about them. Actually, most of governmental institutions here and worldwide are still fairly tight-lipped about properties of those compounds and research. And most, especially initially, most information came from this book that is still available on Amazon, written by Bill Mirzayanov, a Russian dissident and chemist who worked on development of these agents in the 1970s in former Soviet Union. And he described both structures of compounds, events that led to their development, and people involved in those events. And that book turned out to be correct for most of the time later. So a mechanism of inhibition of these compounds is a covalent attachment to active serine of acetylcholinesterase. Here you see a little cartoon representing interior of the active center of enzyme, and here you see actual X-ray structure of serine buried at the base of active center gorge. So one of these organophosphates will come and form reversible complex with the enzyme, because it actually mimics a substrate for the enzyme. So this is Michaelis-Menten type complex that will, when electrophilic phosphorus comes in vicinity of nucleophilic oxygen of serine, form a covalent bond. And now we have intermediate in hydrolysis of this enzyme, of this compound by enzyme, that is extremely stable. It's not irreversible, but it's so stable that it can lead to death, basically. So in this case, it is unique property and that is why these are so difficult to treat. Unique property that they are fairly large, larger than any other nerve agent or pesticide in use, organophosphorus-based pesticide. They fill completely the lower part of the active center gorge of enzyme, and you can see it on the left. So with these two different ways of representing enzyme structure, Nanome has first question for you, and that is, what methods do you use to perform visual structural analysis? So we'll wait a few minutes to see the answers. I hope you're selecting one of those options on the screen. Okay, so Pymol is, it looks like, most popular, but right next is Nanome. So yeah, no wonder Nanome is really spectacular here in its features, and I personally prefer Nanome over anything else. So let's move on to treatment of poisoning that Alexei received first in Russia and then in Germany. And this is actually summary published last year in the Lancet by a group of physicians that treated him in Berlin. First few days, it's a little obscure. We don't know quite with certainty what was he given. Most likely atropine. That's a standard therapy in poisoning. And he was obviously put on ventilation. That saved his life, both with atropine. But on third day, as soon as he was transferred to Germany, they determined that in his blood, cholinesterase levels were way too low compared to normal levels described here by this red dotted line. And he was therefore given antidote against OP poisoning. So something of the kind that we want to develop, obidoxime. This is structure of obidoxime. It's a standard pyridinium-based oxime. In the graph, you see activity, a catalytic activity of cholinesterase from his blood in red triangles. It was basically zero in first several days. And blue circles describe effect of antidote at therapeutic dose that it was given, about 100 micromolar effectively ended up in blood. And obviously there was no effect. So it was not effective at all. Question now is, should we give up on oxime antidote therapy, or should we perhaps try to develop something more effective? And I vote for working on getting more effective antidotes, because recovery of catalytic activity of enzyme is the only true remedy of poisoning in this type, in this case. So what obidoxime was supposed to do was to, with its nucleophilic hydroxyl sulfoxime group warheads, nucleophilic warheads, supposed to attack phosphorus in the active center of inhibited enzyme, participate in breaking bond between phosphorus and oxygen, and release active enzyme. And that would help terminate symptoms of poisoning. Now, why it didn't work, we don't know quite, but this is one description of possible reason. This is a Nanome representation of X-ray structure of Novichok inhibited enzyme. There is X-ray structure of it deposited in PDB. And what I superimposed here is another X-ray structure of obidoxime complex with native enzyme. And you can tell already that nucleophilic warhead of this antidote positions itself in quite good spatial three-dimensional position versus phosphorus, and there are no clashes. But trouble will come in the lower right corner part of the screen here. And pay attention to these three highlighted aromatic residues. They are in stacking interaction with pyridinium that carries nucleophile. And they are on the opposite side of phosphorus that is supposed to be attacked. So there is a distraction, stabilizing distraction here that is not contributing to chemical reaction that is supposed to happen. And that is certainly one of distracting factors in action of obidoxine. So instead of seamless attack, here we have distraction by stacking. I tried here to represent three-dimensional structure of enzyme and inhibitor and reactivator, but now Nanome has a second question for you, and that is on scale 1 to 5, how easy it is for you to navigate 3D structures on a 2D screen. So please check one of the options on scale 1 to 5. Most answers were initially Pymol and Nanome, how easy it is for you to use that software in 2D screen. We'll wait a few seconds for answers to return. And here's something coming. Well, about 38% responded medium, level of 3 on the scale of 1 to 5. And that is understandable. Every software requires some training, and in particular, when it is not immersive type of software, then it becomes a little challenging to perceive three-dimensional structure of macromolecules. So that's where Nanome's advantage comes. So let's move on to next slide and of my story, and compare what is available in the field of antidotes against OP poisoning. In Germany, it is obidoxine that it is approved. That's why they treated Alexei with obidoxine. That's the approved therapy for it in Germany. In US, it is 2-PAM or pralidoxine in the upper right corner. In the UK and Canada, it is HI6. But all of those antidotes, you will notice, are pyridinium-based. They are positively charged, and their nucleophilic warheads, highlighted here in red, are relatively close to pyridinium entities. That is not so good news because pyridinium rings like to stack to aromatic residues in proteins and in this enzyme as well. And this enzyme actually has active center gorge that is lined with aromatic residues, tyrosines, phenylalanines, tryptophans. So there is quite some distraction to placing nucleophilic entity close to phosphorus. Now, what we are trying to develop is something slightly different, and that is this structure in the highlighted in orange at the lower part of the screen. LG703 is one of the leads that we are working on now. It has been proved good in vitro, in test tube, reactivation of inhibitor enzyme, and now we have studies ongoing right now in laboratory animals. So what is different here is that we don't have permanent charge. We have protonated nitrogens in the center, but the other, and that is the anchoring point. So unlike pyridinium, this type of heterocycle will position antidote in the enzyme active center, and then nucleophiles are positioned at little longer distance and in arm that is conformationally more flexible. Why is that important? Because every organophosphate has a different structure. So poisoning by VX, poisoning by sarin, poisoning by Novichoks will all yield different type of targets that we have to attack with our antidote to recover activity. And some flexibility in conformation here is really important in order to circumvent that structural diversity. So there's a number of structures of OP inhibited enzymes deposited in PDB, and in the past 2 years during this pandemic period, group of students and I used to meet regularly, weekly, in VR, actually, using Nanome. And that was really perfect tool because we were all at our homes, but then at the same time joined together in the same VR room and analyzing those PDB deposited structures in order to figure out how to refine this lead structure to fit better our targets. And this is one feature that is important, and that is that this molecule of antidote is actually not symmetric at all the way it looks in the screen here. These are two X-ray conformations from X-ray structures of both ovidoxime and LG703, but in particular LG703, when we compare it to structural formula, has two arms bearing nucleophilic groups in a different conformation. So that's really good news, because anchoring points and only one of two nitrogens in this heterocyclic ring will get protonated. So that will be main anchoring point. Those will result in several conformations of side arms, and these are all X-ray derived that you see here. And distance between anchoring point and nucleophilic attack, point of attack, while it is only one distance in ovidoxime and pyridinium oximes, here we have four, at least four in this conformation of compound. 1, 2, 3, 4, and a single oxime molecule can treat several types of OP poisoning. That's important because it broadens its spectrum, and in particular because in cases of mass poisoning, it is frequently, even in single person poisoning, it's difficult to know what organophosphate was used. We could find out that cholinesterase levels are very low, so it's OP poisoning most likely, but what type of organophosphate, it takes time to determine, and one has to act quickly in minutes in order to preserve life. So in this refinement of this lead structure, we used actually Nanome, and we used computational capacities or plugins in VR. And this is visiting student, Fulbright fellow that worked on that. This is her poster that we presented at SOT meeting in San Diego, the Society of Toxicology, that happened about a month here in San Diego. And not only that we used VR to calculate, we used it to present. We had few headsets available by the poster board, and loaded them with spatial recording, each of them. So we were able to hand headset, and each headset by itself would tell story about refinement and what we want to do and what we achieved. It was not the only meeting where we used VR. This is one in Seattle, Washington, a few years ago. This one was in Ventura. And in addition to meetings, I really extensively try to use VR in classes that I teach here at UCSD. And topic of those classes is a structural basis of drug macromolecular action. So these were several classes in the last few years. Finally, question for you based on VR experience here that you noticed is whether you would like to try VR, those of you who already have not, and please respond here. I think it's fairly obvious response, but we'll wait for the response here. Okay, yeah, it was not difficult to predict that this is so attractive and impressive way of structural visualization, macromolecular visualization. And from first-hand experience, I can tell you that over years, I tried to convey message about structural information and activity of different macromolecules in meetings. And here you see where one presentation where I used glimpse stereo, then active shutter glass stereo in another meeting. And finally, more recently, I started with VR in both presentations, but also in smaller group discussions at meetings, because meetings are typically excellent occasion to meet with experts in the field. And here you see one of them, one of former directors of PDB in one of our recent meetings. So in summary of this short presentation, I would like to highlight that this fully immersive way of visualizing macromolecules that is provided by Nanome is really outstanding, and I use it extensively in the research, in developing new candidates based on structure, accelerated refinement of small molecules in teaching, and because it's fairly intuitive to show structures of drugs and their interaction with their macromolecular targets, and in conveying results of our research to colleagues. Believe me, it always draws attention to people in meetings when you show up with a VR headset, and then just that fact gathers people around your poster board, but also it is an outstanding presentation tool. Finally, there is a number of people involved in this research, not only from UCSD, and you see here a few names from UCSD, but also from Oak Ridge National Laboratory, Andrei Kovalevsky, Oksana Gurlitz, so number of X-ray structures for us. Computational chemists Shaolin Chen, Nikolai Fair from Ohio State, and Don Blumenthal, a biophysicist from University of Utah at Salt Lake. And of course, the major source of funding for this was Counteract program of NIH. At this point, I'd like to switch to Nanome and to show you firsthand what it looks like in VR, some examples of cholinesterase, and I'll show up in this window in a second. Let me just put my headset on, and there I am. And what you see spinning is one of those highly toxic molecules of Novichok nerve agent. So that is one obvious advantage of using this type of analysis, because you can have it in your hand and still not get exposed to it. The target is enzyme acetylcholinesterase that you see here, and its active center gorge, as I described, is here. And we see here X-ray structure of inhibited enzyme. What you see is Novichok bound to it. So I'll get actually inside into the enzyme and so now what you see me from the place of Novichok inhibition. And this is the path.
Sorry, Zoran. I think you might just want to do the headset again. The video froze. I think that reset it if you, yeah, took it off and back on.
All right. Any help? Okay.
Yeah, no. Actually, yeah, no. Okay, it's good.
Okay.
I think.
Got it. Okay, so I should probably not talk too much about it. Your in-person test would be best experience of this. I just wanted to show you something else also. What I didn't mention that much was a native enzyme, and it's here in red, so not inhibited enzyme, and its catalytic power. The reason why this enzyme is key enzyme in neurotransmission is its exceptional catalytic power. It is one of most powerful biological catalysts. So what you see here is molecule of acetylcholine there. And I'll put it in motion now, because that's also what you can do. And this little simulation that I put together from X-ray structures, actually we solved several X-ray structures of enzyme in complex with a substrate, shows that those conformations that you see in the gorge are actually X-ray structure snapshots. What is fairly obvious is that path is fairly treacherous and narrow for this molecule, and it has to run in and out at speed like this, which is 100 per second, and 100 times faster than this, because turnover of molecule is 10,000 per second. So one molecule of enzyme turns over 10,000 molecules of substrate every second. This reaction, the chemical reaction, is faster than that. And so it's only limited by diffusion. So ability of enzyme to admit that many molecules is amazing, considering its narrow geometry of gorge. With antidotes, we want to be able to accelerate current reaction. Currently, it is at this rate, which is about 100 per second, but we would want to make that reaction run at least order of magnitude or two faster, because really a rate of recovery of enzyme activity is essential. We don't have to recover 100% of activity. It's enough 20%, 25%, but it has to be fast. Otherwise, the poisoning can become lethal. Okay, thank you very much. Here I'll stop and take any questions, either from VR or from actual reality here. So Steve, back to you.
Okay, fantastic. So yeah, thank you so much, Zoran. I thought that was a really great presentation. So yeah, right now, I believe you should all be seeing the Q&A page here. So I saw that we already got a few questions already coming in, but yeah, if you haven't yet asked your question, feel free to go ahead and post that using the Q&A feature in Zoom that you see here. But yeah, Zoran, maybe we could just kind of start with a few of these. So first off, there was an interesting comment from David Baum saying that it looks like you need a longer linker on the nucleophile so it could reach deeper into the binding pocket to react with the electrophile. This would not have been immediately obvious, but was illustrated really nicely using Nanome. So yeah, it sounds like there's already some good research discussion happening.
I could add here a comment and from historical comments, and that is that initial nucleophiles and nucleophilic antidotes and pralidoxime 2-PAN, the one approved in US is example of that, was developed about 70 years ago, so quite some time ago when structure of enzyme was not known. Development was based on fact that it was cation. So because enzyme geometry evolved to catalyze acetylcholine, which is cation, and then cationic antidote made sense. And it was fairly good until now. Obviously, we know much more about enzyme structure and should be able to utilize that to develop better ones.
Fantastic. Definitely be good for you two to connect offline as well. So we got another question from Ken Jong. I found out some of the crystal structures OP inhibited ACHE and PDB, but there was no crystal structure of the Novichok inhibited ACHE. That's because of it being tightly regulated, and or do you want to address that?
Yeah, it is really interesting for someone doing work in this field how your own colleagues that you know from a long time ago suddenly become silent, stop talking when you mention name Novichok. It's really funny. But in any case, actually there are structures of enzyme inhibited by Novichok's. There are 6 structures in PDB, but you won't find them under, most likely if you use Novichok to search. Try search with A-230, 232, or A234. Those are code names for those inhibitors, and 3 of them have been deposited in their native state, and 2 of them or 3 of them with in complex with another antidote. So yes, there are structures.
Okay, and so Novichok just being the classification of these newcomer nerve agents, but then them having their code names to look up the protein structure.
Yeah. It is interesting, though, that there was no accompanying article to those structures. So typically, when one deposits structure in PDB, there is a adjoining article describing that structure. You won't find that kind of article in this case. There's only structures deposited, which is immensely useful for us working in the field because we can study. But we don't have firsthand description from people who sold structures.
Great, cool. So we got another question here from Christian Kolakowski. What is the best way to measure potential benefit realization to justify the cost and effort to visualize using VR, which I might add that cost and effort is going tremendously down as consumer technology just makes all of this really easy. Using VR, several proteins in 3D simultaneously, any templates used or qualitative assessment of value delivery. So basically measuring the value of using VR. How do you do that?
Yeah, well, historically, as I mentioned, I've been using different approaches to macromolecular visualization. And those of you who are a little older will remember first visualization supercomputers that were put on the market by Silicon Graphics International, SGI, several decades ago. Those were immensely expensive, tens of thousands of dollars for one computer and workstation. These days, one hardware required for VR is orders of magnitude lower. The simplest approach is to use Quest 2, for example, headset, and that costs $300. So compare 300 to 30,000. There's immense value. And you can go to Nanome website, I understand, and try your software for free and see whether you like it or not. So there is this free version of software that you can use to do some initial visualization. And it's really for educational purposes, well worth investment. For me, it was really great. Works great.
Great, great. Thank you, Zoran. This one's also interesting. I personally have spent many hours in VR in a row without discomfort. But Ludo asks, what is the longest session you've had in VR without discomfort?
Um, Actually, I have to report that I had practically no discomfort at all using Nano, which is not true for other VR-based software. We had some projects with NASA, and from a long time ago, I was really impressed by space flight and research. And there is one application on Oculus website that you will find, ISS, International Space Station Explorer. That one is really beautiful if you're interested in that type of visualization, but that one caused me discomfort while I was floating in the space. Nothing like that in Nanome. So of course I overcame that ISS discomfort fairly quickly, but I never had any discomfort using Nanome, and maybe because I had to think too much about structures and I was distracted by topic. So really, those side effects did not come to distract me.
I'd like to think that's a little bit of the way we built the application, that you're not flying through space in a tunnel, and we try to have really high frame rates. So we definitely care a lot about the user comfort because we want you to focus on your molecules and not have to worry about the other things with VR.
Yeah, sure.
Yeah. Great. So, so Zoran, I know that you kind of made your own, plugins before we had the plugin API with your CSV data parser into the PDB format to be able to parse that. But, Miker asks, what are your favorite plugins available in Nanome?
Currently, at this, first of all, I have to say that I've been impressed by the rate Nanome has been able to improve and build this software. And plugins are, at this point, really useful for us to evaluate structures in VR. What that means is both minimization, so we can build a molecule, small molecule in VR and then minimize it inside. We can dock it to a macromolecule using SMINA docking, which is state-of-the-art docking software. We can, for covalent conjugates, and we deal with covalently inhibited enzyme, we can minimize and score. We can score, do real-time scoring. So my favorite one is SMINA at the moment, but all those computational components I use most at this time.
Great, great. And there's also RMSD and chemical properties, and there's a whole slew that you probably use in conjunction with each other as you go. This one's kind of in a similar vein. So how do VR tools fit for visualization of dynamics and differences between two similar structures? This is from Stanislaus Cherepanov. Thank you for asking that question.
Yeah, here I get to use my creativity a little bit in VR, and there are two structures that have to be, or more, to be compared. So, for example, two snapshots in molecular dynamics, or even two enzymes, inhibited and non-inhibited enzyme, or liganded and apo enzyme. And it's useful to blink back and forth two structures in the same orientation, just switch back and forth quickly. And here I create typically animations. So I animate multiple snapshots in as many frames as are needed. Typically for MD, there can be dozens of frames, but for a comparison of structures, 2 or 3 are fine. And those blinking movies really reveal a lot, and especially for VR, where one can vary rendering and display of particular parts of protein. That's really what I like to do.
Thanks, Zoran. All right, so another one from Christian. So we have compared to traditional structural analysis tools like PyMol, how does Nanome accelerate your work? Yeah,
I started with PyMol and similar 2D based software, but you know, at one point I even participated. I had in solving some X-ray structures, and maybe that is the best illustration of comparison. So when solving X-ray structures, one goes from one residue to another residue of protein and tries to fit atoms into electron density. And frequently you will notice those people, structural biologists who do that, constantly move their mouse left and right, left and right. They try different angles of view to a protein in order to mimic 3D. That's the only reason. And so that is the basic difference between 2D based software and immersive VR. You don't have to move it left, right. You are already in the site, and you can perceive atoms around you instead of trying to look for a different angle of view.
Yeah, I remember you getting in there and just saying you didn't realize just like how much space was in the pocket, and it just becomes immediately obvious. You get intuition for the nanoscale, and that's something that nobody has because we don't live at the nanoscale, we live at the human scale. But yeah, definitely great to hear that you're getting a lot of value out of having the full 3D nature literally be in your hand.
Yeah. That's for me, and not to talk about students who get exposed to molecular structure for the first time. That's really impressive effect.
Great. So we have another question here. So what aspects of virtual reality do you find most relevant in your daily research activity? Well,
there is an effect of entering VR that is even not related to structure. Pace of daily activities, or activities daily, have their own pace. But once you put headset on, all of that suddenly stops, and you enter a different reality. So this is like jumping into a pool of water. You're suddenly in different medium. And for me, it works like instantaneous focus on a topic. I am instantaneously focused on my atoms and molecule and side chains of amino acids. So distraction is canceled out. And in addition to immersive visualization, there is this effect of being transported into nano world, actually.
Great. So I remember the first time I came to your office, it was like a history of all the 3D interfaces, different classes and everything. And of course, you had the big TV. But the question here is, what drove you to start using virtual reality?
Yeah. So I have to acknowledge here my former mentor and supervisor, Palmer Taylor, here, because he I was in his lab, and he had this profound interest in molecular structure, and he supported every initiative that I had So I really had open hands to try various kinds of visualizations and technologies until I came up to VR. And I'm sorry, the question about VR was
just what drove you to start using VR? Yeah, how did you get into it?
Yeah, the simple answer to your question could be, I simply got tired of standing in front of that window. And now some people will understand, but let me explain this a little bit. When you use any kind of 3D visualization technology, side-by-side stereo, shutter glass stereo, or anything of that kind, you end up standing in front of a window that has 3D depth cube. You can see molecules behind that window in third dimension, but you can't actually get in there. So I got tired of standing in front of action and not being actually part of action. And as soon as I learned about possibility to use VR hardware to display molecules, and that my first contact was a colleague in Sweden, Jonas Bostrom, who created one of very first softwares for Oculus platform, I jumped to it, and then fortunately, and it didn't work for me, and then fortunately we got in touch. Your group was started developing the Nanome at that point. It was at early stages, but it was so much better, and that's how I started. I was really lucky to have someone nearby, walking distance from the office, that I could go and complain about. I can't see that. Make it visible, make this control better, make it display this surface different. And so, you were very responsive. I have to acknowledge to everyone that those people working in Nanome are really responsive to our comments. So they got something really valuable at this point.
Definitely, yeah. We got our start at the UC San Diego campus, and so yeah, it was just I don't know, 5-minute walk between our offices, and yeah, we had a lot of good back and forth in the early days there. And I think this kind of ties in with Jia Wang's question here. Could you change to the display style in Nanome via menu? So obviously we built the display menu. You kind of have your surfaces, ball and stick model. Yeah, do you want to talk more about the representations that you commonly use in Nanome and switch between?
Yeah, so the representation that really helps a lot and ability to switch it back and forth is Connolly surface type representation. So was that the question? Yeah.
It was just, can you change the displays? Which, yes, you totally can. But then I just asked if you want to talk about your favorite ones and what you use.
Yeah, typically I spend some time perfecting a type of visualization that that is useful for me, because when you open PDB file, when you just download it from PDB, there is a clutter of atoms that are, some of them have no meaning, some are crystallographic artifacts, and so on. So cleaning up all of that and just displaying the ribbon and selected side chains in addition to surface, that is what I typically like the best.
Okay, great. We have one that's a little bit more technical from Professor Nick Turner. Did you find the presence of the recovery compounds affecting the shape of the active site, the gorge, alongside the effect of the compound within, and was this visualized in Nanome?
Yeah, that's a very good question. Actually, this is what it is exactly topic of our interest right now also. The fact is that some of organophosphates change shape of active center of enzyme and make recovery of activity difficult. Once antidote binds, and there are X-ray structures to support that, it will, in some cases, recover original shape of the molecule, but in other cases, there have been structures deposited, for example, with taboon inhibited enzyme. Reversible ligands that are supposed to be antidotes actually stabilize parts of structure in different conformation. For now, we are taking those conformational diversity and trying to study it and understand it with aim. Our goal is to take advantage of different flexibility of different domains of enzymes to address, perfect our antidote structure so that it can avoid or take advantage of that flexibility, either way. So yes, there is interaction. We don't know for sure what it works like in solution, because these are all X-ray structures. They refer to crystal state, but there is crystallographic information that shows change in shape. Yes.
Great, and I think we have time for one more question before we go into the Meet the Speakers in virtual reality, which is highly recommended because seeing it on a 2D screen is going to be limited in any capacity, and obviously hopping into virtual reality with Zoran and I is going to be a much better experience to actually see what's going on. But let's see. So these are kind of related. Do you find Nanome to be more beneficial for design of novel structures or the analysis of existing 3D structures? And there's another one, is designing small molecules in VR easier than 2D? I think they're both kind of related here, if you want to just talk about your experiences.
Yeah, I'm not sure that I picked up all parts of the question, but whether it's more valuable for analysis of existing or design. I would say currently looking at tools that are available, one can actually use it for both. And these computational tools, plugins that are now available are really useful in design of new molecules. So one can really benefit from, I would call it accelerated template-based drug design. It's accelerated because one can get in to that molecule of interest and then instantaneously recognize what fits and what does not fit, what would be beneficial with respect to small molecule structure and whatnot. Our topic of research is a little unique in that we, like many other researchers, we are not looking for good binders. You know, sometimes people use a structure to analyze what molecule will bind best. We actually need to have molecule bound and in proper orientation and distance to a reactive target. So it is basically a design of substrate. And computational part, I would answer here that keeping that in mind, that the design of new molecules is really what makes it what is really advantage of this software.
Great. So yeah, I just wanted to say thanks again, Zoran. This has been an awesome webinar, and thanks everyone for asking some really engaging questions. At this point, we're going to be moving to the virtual reality Meet the Speaker session. We're actually going to be using a different Zoom link because right now this is a webinar Zoom link where only we could talk with you. However, we'd like to get a little bit back and forth engagement. So your two options for joining are to either boot up Nanome, go into Nanome. There's a room called Webinar UCSD. The password is UCSD all lowercase. if you do not have access to a virtual reality headset and would just like to view this in a video format, that's actually going to be using a different Zoom link than this one. So I'll go ahead and leave this up. But yeah, Zoran is hopping into virtual reality now. And we are going to be streaming on that alternative Zoom link for everyone here. But yeah, thanks so much for joining the webinar, and we will see you on the next Nanome webinar.